So at the presynaptic axon terminal for a serotonergic synapse, you're going to find the enzymes that'll basically, take tryptophan and generate 5-hydroxytryptamine, or 5-HT. And then you're going to find that monoamine vesicular transporter. So this is a transporter that's located in the vesicular membranes of all the monoaminergic presynaptic terminals. So for dopamine and norepinephrine, they use the same gene and generate the same monoamine vesicular transporter. It does not care what monoamine happens to be present. However, in a serotonergic synapse, or presynaptic terminal, the enzymes that are expressed in that cell are going to generate serotonin from tryptophan. So that's the one that this monoaminergic vesicular transporter will actually pull in and package in the vesicles, for release. So there's, typically released upon the arrival of an action potential, and then serotonin, there's exocytosis of serotonin into the cleft, along with, whatever other co-localized neurotransmitters are also in the vesicle with our primary neurotransmitter of serotonin. And then it'll stimulate receptors. And again, there are, 15 potential, receptor targets for serotonin, both pre and postsynaptic. The majority of these, 14 of them are metabotropic receptors, linked to the initiation of various biochemical cascades or, the sort of delayed opening and persistent then opening of, specific G protein gated channels, upon the release of the G protein. But what's interesting about this is that the removal process for serotonin is through a selective presynaptic terminal, what we call reuptake transporter. So this is like, serotonin is released from this neuron into the cleft, and then on the membrane of the presynaptic terminal right here, very small on this pipe cleaner, are going to be reuptake transporters that are selective just for serotonin. So these selective serotonin, reuptake transporters are going to pull the serotonin out of the cleft and bring it into the presynaptic terminal. In the terminal, it's going to encounter an enzyme, a degradative enzyme called monoamine oxidase. It's actually abbreviated as MAO. So MAO will grab onto serotonin, but, given its name, you should know or, suspect that MAO will also grab onto norepinephrine and break it down. It'll also grab onto dopamine and break it down. It's a monoamine, degradative enzyme. It's not specific at all. But that reuptake transporter that's, based in the, that's found in the presynaptic terminal membrane, that is selective for serotonin. And this is true of all the monoamines. So for the dopaminergic presynaptic terminals, you've got, inside the dopaminergic cell, you're going to have the specific enzymes that'll take tyrosine, the raw material for the manufacture of dopamine, and convert it into dopamine. And then you're going to have a, very open, monoaminergic vesicular transporter, in the vesicle membranes. It would take in any, monoamine, whether it's dopamine, norepinephrine, or serotonin. But in the case of a dopaminergic neuron, what's being synthesized is dopamine. So dopamine gets packed in by that very, open monoamine, vesicular transporter that would take in any monoamine. But in the dopaminergic neuron, it'll take in dopamine. Dopamine is released upon the arrival of an action potential into the cleft, and then dopamine will encounter a selective dopamine reuptake transporter in the presynaptic membrane that'll pull dopamine into the cleft. And then it will also encounter that, very nonspecific monoamine oxidase, the degradative enzyme inside the presynaptic terminal. Then in the case of a dopaminergic neuron, we'll encounter dopamine and break it down. And the same is true of norepinephrine. Norepinephrine are noradrenergic neurons, right? They're going to synthesize the genes for the synthesis of norepinephrine from the raw material tyrosine. It'll make dopamine first, and then it'll turn that dopamine in a noradrenergic neuron into norepinephrine or noradrenaline, right? And it'll pack it into the vesicles again with a very, non-specific monoamine. It'll take any monoamine, vesicular transporter to pack in, but in the noradrenergic neuron, it just made norepinephrine, so that's what it encounters, and that's what it will store. It'll transport and store in the vesicles, for release upon the arrival of an action potential. And then, when the norepinephrine gets released, it gets reuptaken, and that is literally the way they talk. Reuptaken through a selective norepinephrine presynaptic reuptake, transporter back into the presynaptic terminal of the noradrenergic neuron, where it'll encounter again monoamine oxidase, that enzyme, that's expressed in all the monoaminergic neurons, whether they're dopaminergic, noradrenergic, or serotonergic. It'll break down any of them. And in the case of a noradrenergic neuron, it'll encounter the reuptaken norepinephrine and break it down. So the selectivity comes in the reuptake transporters, these presynaptic transporters that are selective, for dopamine, serotonin, or norepinephrine. And that allows for, specific drug development. And there's a whole class of drugs. Again, in the psychopharmacology class, we go into much more detail on this. But I know many people have heard of what are called selective serotonin reuptake inhibitors, right? Or SSRIs. And they'll act in the cleft. They'll bind to the selective serotonin reuptake transporter and prevent the reuptake selectively of serotonin after the release of serotonin, upon the arrival of an action potential in a serotonergic cell. Excuse me. And what's interesting about that is that will do the immediate effect is to increase the amount of serotonin in the cleft and the amount of stimulation of all of those serotonergic receptors, including the 14 metabotropic receptors that'll set off new biochemical cascades or enhance certain biochemical cascades and will reduce others. It'll change the sort of chemical network connections between activity at the synapse and the genes, aspects of gene expression and protein synthesis. So there's a lot of very interesting things about SSRIs and other drugs that target, these serotonin receptors and serotonin transporters. They will result in behavioral changes in some cases, actually, like relief of major depression, for example. But oftentimes it's after several weeks of treatment. So that's actually really fascinating in terms of it sort of suggests that there have to be kind of network changes in gene expression and the growth of specific connections. Actually, there's been some really interesting work on the sort of encouragement of what's called neurogenesis, the division of neuronal stem cells in the brain and the migration of those stem cells to sort of build up structures like in the medial temporal lobe, like in the hippocampus and areas. So there's a lot of complexity in what's happening with, blockade of these reuptake transporters that are selective for serotonin. And in other classes, we go into much more detail about that. But I just want you to have a good sense of, what is the synthesis, the storage, the release, and in the case of the monoamines, the reuptake and removal, basically of these neurotransmitters that are so important for alertness, arousal, that modulate lots of aspects of, brain function, and in the case of serotonin specifically, that are really critically important for psychological health and mood.